Hello and welcome to Teach Me in 10. Today we're joined by Kevin Murnan, a field application scientist and qPCR applications lead at Bio-Rad. In this episode, Kevin will guide us through the key steps of qPCR gene expression analysis, helping you get from raw data to meaningful results in just 10 minutes.
Hi, I'm Kevin Murnan, a field application scientist here at Bio-Rad, and I'm going to be showing you how to analyze qPCR data in CFX Maestro so you can ditch Excel, gaining reliable and visual data without the hassle of spreadsheets. During this short video, you will learn how to set up a plate map, labeling target genes, technical replicates, standards, and node template controls. Doing so will allow data analysis to be a breeze with automatic calculations determining reaction efficiency, generating bar charts for gene expression analysis, and even performing powerful statistical analysis such as ANOVA. So the first thing that we want to do when we open our CFX Maestro software is define our plate map. So we can go ahead and specify a user-defined run, and then within the run setup, we can hit plate. Let's create a new plate map from scratch. The first thing that we want to do is choose our scan mode. So we can choose either cyber or all channels if we're using probe-based chemistry, for example, with multiple fluorophores. Let's go ahead and focus on cyber for now. I want to highlight every single one of the wells and specify that we are going to be analyzing data within them and that we have loaded cyber. So first thing that we'll do is we'll just choose a sample type of unknown, which we can change later, and then we're going to load cyber within each of these wells. Next, let's actually define an experiment. All right, so imagine that we are working on an experiment where we have a wild type cell line and a mutant cell line, and we want to look at the fold change of gene A, and we will use gapDH for normalization. Let's go ahead and set that up. So let's have rows A and B. Our biological group can be the wild type cell line, and then C and D, our biological group can be our mutant cell line. Okay, let's specify that A and C, I'm holding control to highlight these. Our target is gene A. And then for B and D, we can specify that our target is gapDH. This looks great. So right now, we have two rows that are wild type, two rows that are mutant. In one of these rows, we have gene A as our target. And in one of these rows, we have gapDH. Same with our mutant. Let's let the instrument know that we are setting up technical replicates. So we can highlight all of these wells, go over here and click technical replicates. All right. Let's say that our replicate size is 3. We can go ahead and tell the instrument whether or not we have loaded our replicates horizontally or vertically. Let's go ahead and keep it horizontal. That looks wonderful. Now we can see that we have technical replicates going from left to right in triplicate for each one of our samples. And we can even specify our samples if we so desire, such as, for example, highlight these and type in sample A. Highlight these and type in sample B, and so on. We can go a few steps further as well. Let's say that we have a standard dilution series on our plate. Okay, we can choose the sample type as standard. We can choose our target name as, let's say, gene A. We're testing the efficiency of this. Let's go ahead and specify that we have our technical replicates in maybe duplicate. So we're doing 6 dilution points in technical duplicate going from left to right. And we can specify our dilution series by clicking here, saying, yes, we are going from replicates 1 to 6. Our dilution factor is 10, and we're decreasing from left to right. And we can change this accordingly. That's great. Now the software can calculate our reaction efficiency. And if we want, we can do some additional things, such as choosing our node template controls, positive controls, negative controls, and so on. And if we don't want to take a look at some of these wells when we're analyzing data, because we didn't load anything in them, we just clear the wells. And one last thing that we can do is we can actually go ahead and show our technical replicates, if we so desire, which is a really quick and easy way to look at our experiment. Speaking of our experiment, if we want our data analysis software to actually calculate the fold change for us, we'll just click Experiment Settings. We will choose our reference gene, in this case GAPDH, and then for our sample and biological groups, we'll just choose our wild type as the control group. That's it. Now, when we take a look at our data, we'll be able to determine the fold change and any sort of associated statistics with it. Speaking of data, the following data were generated from a previous run and provide us an example of the ease for data analysis. Okay, so we have a wonderful looking bar chart in our gene expression area. But let's actually begin from left to right as we go across this, beginning with the first tab, which is quantification. On this particular plate, we're looking at 3 different cytogreen targets, okay? Actin, IL-1 beta, and tubulin. And we can go ahead and get our amplification curves, which are in log scale. We can look at them not in log scale if we so desire. What I really like about this software is that we can hover over anything and all of these different charts are connected. So right here we have our amplification curve, which is connected to our reaction efficiency, where our unknowns are plotted as x's and our standards are these circles. Okay? The software, if we set up our standard deviation series, will automatically calculate the reaction efficiency. Additionally, in the bottom right, we can get a sneak peek of our data, such as our CQ values. Anything that you see on the software, you can right-click and save as or copy. Hopping over the quantification data, we have our charts that traditionally people like to export these by clicking right-click and export to Excel or CSV. But one thing that you could do if you've set up a gene expression experiment is simply hop over to the gene expression area. As long as in the experiment settings, you have specified your reference gene and you have specified your control, the data analysis software will generate charts for you. Now, not just will it generate charts for you, we can even annotate statistically significant fold changes if we so desire, and we can even choose our confidence interval or the standard error of the mean or standard deviation for our error bars. I tend to like standard error of the mean. It looks pretty good. Not just that, we have our clustergrams, scatter plots, volcano plots, heat maps, and even ANOVA. We can see the difference between various time points on this particular qPCR data file. And not just that, we get our p-value turkey. I'm hungry for turkey. We can also take a look at our melt curves if we so desire. We can look at individual genes by getting rid of the others. And what we like to see is a single melt peak, indicating that, yes, our assay is specific. Finally, we have our quality control tab. Anything that breaks these built-in quality control parameters will be highlighted for us. For example, I can see that actin has broken our reaction efficiency. It's less than 90. I can come over here to the quantification and see, oh yeah, actin's efficiency is 88.2. Similarly, we can change these parameters if we so desire, okay, by just clicking and altering them if need be. But anything that breaks these parameters will be highlighted by the software. And not just that, we can exclude them from our data analysis if need be. And finally, within the data analysis table, we have the flexibility to customize this as we so desire. So for example, if we want these charts to be different colors, we can definitely do that to make it a little bit easier on the eyes if we so desire. And with that, I believe that you are more than equipped to use the CFX Maestro software. Thank you for your time, and I hope you have a wonderful day.
Thanks for watching this episode of Teach Me in 10. To learn more about CFX Maestro software and qPCR data analysis, check out the resources linked in the description. And if you enjoyed this episode, don't forget to like and subscribe to stay up to date with all of our latest releases. See you next time.